right. Nice to see y'all. Today's test, I think it's calculus right now. So here is the last week of AP exams. If you've got a morning one, you're supposed to be there at 7:30. If you got an afternoon one, you're supposed to be there at like 11:45 at the latest. Little tip, if you get there earlier, for example, our biology test on Thursday, it says get there at 11:45, but Miss K was basically saying if you got there at like 11:35, we could probably get it in, and we will possibly be done before 4 o'clock. So if you've had 8th period off for too long or something like that, you might forget the difference between traffic of leaving the school at like 3:55 versus 4:05. The idea is if you guys start signing in and everything at like 11:35, they can start reading all the general instructions, and if they get started at like noon, then you'll be done. Or 12:05, 12:10, something like that, you'll be done, and you'll be able to leave, and you'll be able to beat the crowd. If not, then we'll be finishing about the time that school's out. So if you can get there a little bit early, great. Don't forget, and I'll announce this next time I see you guys too, but don't forget on Thursday, if you're taking the AP Bio test, it's an odd day. So you go to first period, you'll go to third period, and then instead of going to your 5th period, you'll go to A lunch. And then from A lunch, you'll have like 25 minutes, maybe 30, and then you can get down there at like 11:35 or something like that and take the test and go home early. So that's always nice. Any questions about AP tests? Cool. And then next class, let's see, Wednesday morning, it looks like is French. Probably not many taking that, but world history. So some of the sophomores are probably taking that one. So some of you guys, I won't see you. So good luck then. On Thursday for the AP Bio test, if like on your way to A lunch, you want to drop off your bags and phones up here, it's supposedly faster for you to get out of here if you leave your stuff here. If not, what happens? I proctored the environmental test and the psych test. If not, what happens is they'll be like, all right, you guys are dismissed. You go to leave, and all the proctors, they have to like count up all the materials, get all the crap together. Then they go and have to lock them up. Then they bring out the cell phones to pass out. So even though it seems like coming up here, grabbing your bag and cell phone, and then leaving would be slower, it seems like it's actually faster. So on Thursday, go to first, go to third, stop by, drop off all your stuff here, go out, have lunch, go take a test, and then you can go. If you were able to drop off, I don't know like if they give you crap about leaving the school and coming back, but if you went and put your stuff like in your car, then that would be even faster getting out of here. So if you have keys with you, make sure that you just put your keys at the front or the back of the testing room, just like where you guys will put like snacks and a drink or something like that. Just don't have them on you. Nobody's going to care unless like, I don't know, you got like a garage door opener and when the battery runs low, it beeps. If it beeps, then your test won't count. But if you have your keys up at the front by like your snack and your water and it beeps, probably nobody gives a crap. So any questions about that? All right. Back up here. A couple things before we do some chi-square review. Next class is student choice. So today is the 13th. Next class is student choice. So I've got one thing that I want to do, and then after that, it's student choice. So think about it, and here in a second, we'll just kind of vote on what you guys want to review. And don't forget, on AP Test Day, bring number 2 pencils, blue or black pens, and a calculator. What topics do you guys want? So today we'll be doing chi-square and some Hardy-Weinberg, so no need to request chi-square. We're going to do that in just a second. So what topics do you want? Okay, cellular respiration. What else? I can walk you through just to inspire you. So you got biomolecules. That's like proteins, lipids, saturated, unsaturated, double bonds, things like that. Are they liquid? Are they solid? Got enzymes, enzyme inhibitors like competitive and non-competitive. We've got like levels of protein structure. That was like primary, secondary, tertiary, quaternary. We've got cellular respiration and photosynthesis. We've got cells like organelles. And then we've also got cells like all the membrane, transport, receptor type of stuff. We've got cell cycle. That was like G1, S, G2, mitosis, proto-oncogenes, tumor suppressors, that kind of thing. You've got Mendelian genetics. That's like, I call it Mendelian genetics. It's actually like, there were like 9 or 10 things where it was like Mendel's rules and then ways of breaking them. But that's all the Punnett square type stuff. We got molecular genetics. That's where you get into like DNA, RNA, transcription, mRNA, tRNAs, codons, anticodons, that stuff. You got ecology. You got evolution. That's right. I think I got everything. I might have missed something in there. Yeah. Did you do enzymes? Huh? Did you have your hand up earlier, Cindy? Photosynthesis. Cell cycle. All right, that's actually plenty. So if there was more and more stuff, then I could kind of like do some things in more detail or some things in less or whatever. But we got, sounds like cellular respiration, photosynthesis, enzymes, and cell cycle. Anything else? And we'll be doing some chi-square and Hardy-Weinberg here in a minute. It can. So I'll basically, cell cycles like G1, S, G2, and then like mitosis, and then do you want mitosis also? Okay, so yep, it'll include mitosis too. So prophase, metaphase, anaphase, telophase, that stuff. All right, so that's what we'll be doing next time. And the only assignment that's still out there is the ultimate review. So if you haven't turned in your second installment of ultimate review, it's not due until next time. But if you haven't turned it in, I'll only say that because some of you guys turned it in like weeks ago. So if you haven't turned in your second installment of the ultimate review, it is due next class. If you're in, say, world history or something like that, and you want to email it to me, if you want to drop it off like after the world history test or something like that, that's fine. So if you're like in the middle of an AP test on the morning of the 15th or something like that, I'm sorry, the morning of the 14th, then don't, I'm sorry, the 15th, sorry. Yeah, anyway, if you're in the middle of the AP test and all of a sudden you're like, crap, I forgot to turn it in, just drop it by like afterwards or something like that. My main goal is that I have it before the AP Bio test for obvious reasons, so that it's a review. Other than that, I think we're good. The only major announcement that you guys missed, or that 3 of you guys might have missed, is for the AP Bio test on Wednesday, instead of getting there at 11:45, it seems like if we get there at 11:35, we'll be done and leaving before 4 o'clock. So we think that we could like have you guys like walking out of the building at like 3:50, 3:55. If instead everybody shows up at 11:45, the test is going to be over at about the time that like everybody's leaving the building. So 3:55 traffic versus 4:05 traffic is very different. So if you can get there at about 11:35, that'd be great. All right, and you'll take first lunch on that day. So you got a first period, third period, and then instead of going to fifth, you'll go to A lunch and then the test. All right, any questions about stuff? All right, well, I had a question for you. It's a chi-square question, and you might have this one. Let's see. It was on the back of another question I had given you. So if you have the question with all the faux stuff, it was like faux 81, and there was like little kind of triangles and circles and ovals and stuff all over the place. If you look at the back of it, there's a chi-square problem. And the chi-square problem looks like this. So if you have it, if not, I've got some extras, but take a moment, see if you can find it, save a little bit of paper. But if not, I've got some extras. So it has like faux stuff on the front. Yeah, that's it, right there. That one. And then flip it over. That's the chi-square problem. It's got faux stuff on one side, and it's got this chi-square problem on the other one. And I gotta be honest, I can't recall if the faux stuff was something we did in like an after-school review or if it was like in class. So it might be that some of you guys have never even seen this faux thing. It was in class? Right. Thank you. All right, if you don't have it with you, no worries. I've got some extras, so raise your hand and I'll bring it around to you. There you go. Anybody else need one? Cool. All right, so this is the hardest chi-square problem that I've seen on a biology test. It's older than you are. It's from 2003. It was the second year that I was teaching AP biology. And a couple of things. They give me this, and then they also give me a formula sheet. So this is what your formula sheet is supposed to look like this year. It's like the 2004 version. And so on the front, they've got the chi-square equation, and then they also have like the chi-square table. And so I'll have the table up here in a second. The table's kind of nice. It's not as big as the one that you guys are used to having to kind of manage. There's only 2 p-values, so I do have to know like what p-value to use, and I do have to figure out degrees of freedom. But it's not like that giant chi-square table that we've used in the past. And other than that, I think you're good to go. I'll go ahead and read it. It's like story time. So let's see. In fruit flies, the phenotype for eye color is determined by a certain locus. Remember, locus is location. So a locust is an insect, but a locus or loci is like locations. So if there was multiple locations, they call that loci. Go figure. Capital E indicates the dominant allele. Lowercase e indicates the recessive allele. The cross between a male wild-type fruit fly and a female white-eyed fruit fly produced the following offspring. There you go. That's the F1 generation. It then says the wild-type and white-eyed individuals from the F1 generation were then crossed to produce the following offspring. There you go. So we got the parents up here. F1, that's the kids. F2 generation, that's the grandkids. All right, so part A, determine the genotype of the original parents. That's going to probably be a point. And explain your reasoning. That's probably going to be a point. You may use Punnett squares to enhance your description, but the results from the Punnett square must be discussed in your answer. That's pretty lengthy, so maybe part A is worth 3 points or something like that, I'm guessing. Part B, use a chi-square test on the F2 generation data to analyze your prediction of the parent genotypes. Show all of your work and explain the importance of your final answer. All right, so I'll leave you guys to it. We will come back and do part C, but part A and B is all the like Punnett square and math stuff. So give it a shot. I'll hit pause. And that way we don't mess up something on B, because on this one, part B depends on part A. So when I look at A, it says determine the genotypes of the original parents. So by the way, if you're watching the video at home, this is 2003, number 1. Okay, so the genotype of the parents. So what do I notice that's kind of odd about the kids? If I'm trying to figure out how the Punnett square works. Yeah, so basically there's 101. Now if I read part C a little bit later, they tell me the brown-eyed female, is a result of a mutation. So we never predict that. We're never like, I predict there's going to be a random mutation. So I've got 45 and 55. So I've got 100. And out of those 100, none of the wild type eye color is male. And none of the wide-eyed ones are female. So what's probably a pretty safe bet on this? What kind of inheritance is this? Sex-linked, because it seems like it depends on whether I'm male or female as to what kind of characteristics I'm going to get. So, all right, if we think it's sex-linked, then the next thing I have to do is try to figure out, like, all right, they told me to use the letter E. So if it's sex-linked, then I've kind of got a couple of options. So here's one possibility. This will be our other possibility. So on one of them, maybe, it says I've got male wild-type fruit flies and female white-eyed. So I don't know what's dominant. So I don't know if white eyes is dominant or if red. Turns out their wild-type eye color in fruit flies is red. So I basically am like, all right, is it this? Is capital E wild-type and lowercase e is white eyes? Or is it this? Capital E is white eye. Lowercase e, wild type. So I can run these two Punnett squares and see if one of them seems to fit and see if one of them seems to not fit. It looks like, bless you, that the outcome is pretty close to 50-50. So I've kind of got like wild type females, white-eyed males, about 50-50, and then none of these. All right, so let's see. If capital E was wild type, I know that the male is wild type, so the dad. So these are the parents. That's mama, that's papa, and then these are the F1s, their kids. So you always have like parents, their kids, grandkids, great-grandkids, and so forth. So F1, F2, F3. All right, well, if the male has wild-type eyes and capital E is wild-type, then I know the male has to be this, capital E. And then it says the female has white eyes, so she would have to be little e, little e. All right, cool. We'll come back and see what the kids are. Actually, we'll see what the kids are now. So if this happens, then it looks like all the girls have wild-type eyes. They would all be capital E, wild-type eyes. And that's consistent. All the girls have wild-type eyes. Then we look at the boys. And it looks like all of the boys would have white eyes. They'll get little e from mama. And that's what we find. So this is 55-45, not 50-50. But that's pretty close. That's likely due to chance. So it seems like that Punnett square can work. Let's see if the other one can work also. If the other one can work, then we're like, who knows? So now, let's say that white eyes were dominant and wild type is recessive. All right, I know the dad for sure has wild type eyes. So if wild type was recessive, then dad has to be this. That would have to be dad. Mom. It tells me that mom has white eyes. If white eyes were dominant, then mom has to have a capital E. And then the other one is a question mark. It could kind of go either way. Right? All we know if she has white eyes, if white is dominant, is that she would have one capital E. All right, well, let's see how that works. If that's the case, then I'd have a girl right here with white eyes, a girl over here that's kind of an unknown. I'd have a boy over here with white eyes. And then a boy that's unknown. I'm like, all right, well, I'm not even sure what it means. However, I noticed this. This means that out of 4 kids, males, like 25% of them, should have white eyes. All right, it's more like 55-55, so, or 55-45, so maybe that works. And then females, 1 out of 4 at least, maybe even this one, but 1 out of 4 for sure should have white eyes. But none of them do. So we were expecting like 25 to have white eyes, but instead 0 do. So that means that one's probably a no. Right? The odds of that. This one works, 50-50, that's pretty close. This one over here, for sure a 1/4 of the girls should have white eyes. Not to mention we don't know that and we don't know that. So that one's probably no good. This one is probably true. So if it says determine the genotype of the original parents, I would say something like this. I'd have my Punnett square there, and I'd say parents. We'll say male. This. That's the dad right there. And then the female is that. She has little e's. And then for my explanation, I'd have my Punnett square, and I would essentially say, hey, if these are the parents, then about 50% of the girls, about 50% of the kids are girls, and they are all wild type, bless you, and all the males, about 50% of the total, would have white eyes, and that's pretty consistent with what our data shows. So that's what I would say. Right, now that I've got that, then what they do is they say we take the wild type and white-eyed individuals from F1. So we take some of the bros and some of the sisters, and we mate them over here, and these are their kids. So they're flies, they don't know any better. But we mate them, and we run a chi-square, and we're trying to see, hey, is that chi-square, like what does it say about the data? So give it a shot. If you haven't done the chi-square yet, try it. So I know it's not this Punnett square. I can pause this. And so in part C, since that's a mutation, we don't really predict for it. And so I'll have 1, 2, 3, 4 fractions. What I notice is that anytime I do a chi-square problem, I use the actual numbers. I don't use percentages. And if you just want to accept that, cool. But for mathematical reasons, the way the equation is set up, it actually changes your outcomes based on how big your sample size is. So for example, if I made these, if I was trying to figure out if I reject or fail to reject my null hypothesis, and I use these numbers, 23, 31, 22, 24, I might get reject or fail to reject. If instead that was 2300, 3100, 2200, 2400, I might get a different answer. Because if I have bigger numbers, I'm more certain about my answer. So anyway, always use real numbers. Don't use percentages. However, it's kind of convenient. Those 4 add up to a perfect 100. So what I'm thinking is this. If I've got my parents right here, that's mom and dad. I'm sorry, that's, yeah, mom. So that's a parent, and that is a parent. And then all the ones circled in brown here, that's the F1. So now what I'm supposed to do is take the wide-eyed males from F1 and the wild-type females from F1, breed them together. So I'll have one of these ladies with one of these dudes. And so I expect a girl with wild-type eyes. I expect a girl with white eyes. I expect a boy with wild-type eyes. And then I expect a boy with white eyes. So I expect like a 1 to 1 to 1 to 1 ratio. A fourth, a fourth, a fourth, a fourth. Well, I've got a total of 100 kids. 23 plus 31 plus 22 plus 24 comes out to a perfect 100. So that means I expect 25, 25, 25, 25. So I've got my observed and my expected. My expected is 25, 25, 25, 25, like that. My observed, they always have to give me my observed, or at least give me enough information to where I just figure it out. In real life, in a real experiment, my observed would be like my results. I run the experiment, I breed all these flies. After a few weeks, I've got my results. But if they ask me a question, I don't get to do the lab, so they just have to give me the observed. So I observe 23, 31, 22, 24. 23, 31, 22, 24. So now I'm going to have 4 fractions. How many degrees of freedom am I going to have? 3. How did I decide I'm going to have 3 degrees of freedom? Yeah, 4 outcomes minus 1. So whenever I start using my chart here in a minute, my chi-square critical value chart, then I'm going to use 3 degrees of freedom. Is knowing that I use 3 degrees of freedom and how I figured that out, would that probably be worth a point somewhere, do you think? Yeah, so I should write it down. So anytime, now that you guys have looked at my grading rubrics and stuff, anytime you think you have something that's worth a point, make sure you write it down and explain it so we can give you the point. If you just do something like this, if you're like, 4 minus 1 equals 3, they're going to be like, yeah, true. 4 minus 1 is 3. That's not enough to be like, well, it's 4 total minus 1 is 3 degrees of freedom. But you just said 4 minus 1 is 3. You just wrote down random like math graffiti. So somewhere in there, explain like, well, I have 4 possible outcomes. Minus 1 gives me 3 degrees of freedom. All right, so let's plug this in here. I've got fraction number 1. I've got observed 23 minus expected 25 squared over 25. All right, cool. I've got fraction number 2. 31 minus 25 squared over 25. That summation, that sigma, means I'm going to add these together. Cool. Got my third fraction. Observed 22 minus my expected 25 squared over 25. 22 minus 25 squared over 25. And my last one. 24 minus 25 squared over 25. So back in 2003, it's different now. You guys can use calculators now. You get a formula sheet. Back in 2003, there was no formula sheet and there was no calculator. It was actually kind of nice not having calculators because that means that they would give you like pretty easy to work with numbers. You didn't start getting like 7 decimal places to the right or some sort of bull crap like that. So this is actually something I can do in my head. So 23 minus 25 is 2. 2 squared is 4. all of these have the same denominator, so I can just like add all this stuff up. 31 minus 25 is 6. 6 squared is 36. 22 minus 25 is 3. 3 squared is 9. And I do realize that's technically negative 3, but I don't really keep track of the negatives because I know I'm just going to square them away. 24 minus 25 is 1. 1 squared is 1. And so 4 plus 36 is 40, plus 9 plus 1. That means I got 50 over 25. So my chi-square was 2. All right. On the chi-square math, in general, the math itself is about half the points, and interpreting the math is about half the points. So if I forgot how to do chi-square, or I ran out of time, I've said this multiple times, but just throw a number up there, and then if you can analyze it correctly, you can probably still get, like, say, 2 out of 4 points. So what do I do with this number 2? Chi-square equals 2. What do I do with that? What about the chart? Some of you guys went. Yeah, so I've got 3 degrees of freedom. Do I use 7.8 or 11.3? 7.8, because I use a p-value of 0.05. I have 3 degrees of freedom. I know that because I have 4 fractions minus 1. So my number from the chi-square table, my critical value is 7.8. So I'll say 2 is less than 7.8. And so what does that mean? Like, what do I write? There's a sentence I write after that. I fail to reject the null hypothesis. If my number on here, are you in stats? Okay, let me do something for you then. This will help. Because at first, that's going to seem like this contradicts what your stats teacher said, but it doesn't. Let me get the big table up here, so it's more obvious with the big chi-square table. All right, so there's 2 ways to look at this. There's the way that we often do in biology, and then there's a slightly different way that you often do in stats. They're both saying the same thing. So my number is 2, and I'm comparing it to, let me flip over to this camera. All right, so we had 3 degrees of freedom, 0.05, so we're at 7.8. So there's 2 ways to look at this. I can say my chi-square number is 2. That is to the left this way. That's less than 7.8, and if it's to the left, it's probably all the way right about there, then I would say I fail to reject my null hypothesis. In a stats class, what they'll often do is they'll say, all right, your number was 2. About what p-value is that? And you'd say, like, oh, that's about 0.5. Because my p-value is 0.5, that's higher than 0.05, so I fail to reject. So what's weird is, as I go to the left, if you've not had stats, learn it this way probably. If my number is less than this, then I fail to reject. If I'm in a stats class, I'll come back and I'll say my value is 2, so that means it's around 0.5, and that means my p-value is higher than 0.05, so I reject. But as p-values go up, going to the left, my chi-square numbers go down, 5, 4, 1, 0.2, 0.0. So you can either speak in p-values and say I've got a higher p-value, so I reject, or you can say I've got a lower chi-square value, and so I have a higher p-value, so I fail to reject, or I have a lower chi-square value, so I fail to reject. and you can say it either way. You just have to be clear. All right, so if my number was bigger than 7.8, then let's say it was 8 or 10 or 1,000, then I would say I reject my null hypothesis. And that's all we would have to say in a biology setting. In a stats class, you might have to add more. You might have to say, like, I failed to reject my null hypothesis. Any difference from the expected is likely due to chance or something like that. So yeah, if I can tell you how many degrees of freedom there are, and if I can compare these two numbers and like make the right call on whether I reject or fail to reject, I'm going to probably get like half the points. So even if I can't do the math, I can get half the points. All right. As far as doing a chi-square on a free response, it's very hit or miss. I would say it's kind of unlikely. Interpreting chi-square, like in the multiple choice, is more likely. So if they just gave you some numbers and said, interpret this, that's what you're more likely to see. Or on a free response, write a null hypothesis. A null hypothesis is just always what you expect. So they might say something like this, hey, do a test on the F2 generation, blah, 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 write a null hypothesis. You'd say, well, if I cross if I look at the F2 generation crosses from this brother and this sister or whatever, then I expect 25 of these, 25 of these, 25 of these, 25 of these. So my null hypothesis is just write a sentence explaining your expected. Over here, with these parents, my null hypothesis would be, I expect all the girls to have wild type eyes. I expect all the boys to have white eyes. So my null hypothesis is just always, what does random chance predict? Punnett squares tell me random chance. If I've got a Punnett square and I've got mama and papa, what's the likelihood that you have a boy? Well, random chance. So the Punnett square says 50-50 for boys versus girls. So you can be like, girls run in my family. We don't have a genetic reason for that. So we think that that's just chance. You're like, but I'm the girl in my family and I have 3 sisters. Okay, but if you start looking at like births in the United States, it's shockingly close to 50-50. So your family might be like all boys, but somebody else is all girls, and overall, it's 50-50. So, all right. Questions on chi-square? Okay, don't forget, if you end up with one of my calculators, the square root button, you push the square root button after the number. So you can always figure this out by just trying something. Like, I know the square root of 9 is 3. But if I go square root 9, nothing happens. If instead I go 9 square root, it says 3. So that's how I use this calculator. This calculator does not have a squared button, so you just multiply the number twice. So if it's like 6 squared, you just go like 6 times 6. You might say, did you really have to tell us that? Yes. All right, any other questions? Not that you don't understand that, but I'll have a student ask me every year, where's the square? And then they're like, what do I do? And I'm like, if you think about it, you'll kick yourself in a second. All right, we'll just kind of make it math day. Let's try a little bit of Hardy-Weinberg. So Hardy-Weinberg, let's see, you guys have done number 3 here. You've done 3. I don't think you've done 2. This is not on a paper anywhere, so this is only like on the screen. I believe number 2 is new, and then have we done this one with PTC paper? Does that sound familiar? Okay, so we will do 2 and 4 then. So try Hardy-Weinberg question number 2 up here. I'm going to erase this, so if anybody wants it, then take a picture. But otherwise, I'm going to erase it and put like the Hardy-Weinberg equations up there. So this one says a population, and I guess Microsoft doesn't like that. What's wrong with a population of birds? Is there like a space? Is there like a, is my spacing wrong? The population of birds. A population of birds. Okay, I'm going to say it's just a population. So a population of birds contains 16 animals with red tail feathers, 34 animals with blue tail feathers. Heterozygotes display blue. So what is the frequency of the red allele, the blue allele, heterozygotes, and then birds homozygous for blue? All right, give it a shot on your own with somebody nearby, whatever helps. Oh, and then I kind of forgot to talk about part C on that previous one. We'll come back and talk about part C also. So we'll do the Hardy-Weinberg first and then talk about part C. Pause for a moment. All right, let's have a look. So it says, I've got 16 with red tail feathers, 34 with blue. Heterozygotes display blue feathers. Which number is more helpful? The number with red tail feathers or the number with blue tail feathers? Red. Why is that? Yeah, it's recessive. If I start with 35 animals have blue tail feathers, I know that blue is dominant because heterozygotes display blue. So that's how I know that blue is dominant. Well, if I said that, then I would say something like that is blue, that is blue. I'd have p squared plus 2pq equals 34 out of 50. So I'd say like 0.68. But that's not very useful because I have a unknown p and an unknown q. All right. So what we want to do is we want to start with red tail feathers. These are the only ones with red tail feathers. So I know that blue is dominant. So capital B, little b, big, 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 little, little, little. So I know that 16 out of 50, 16 plus 34 is 50. I always, in Hardy-Weinberg, I always have to get those to frequencies. They're always a 1.00. Chi-square, I use real numbers. So it might be 300 of these, 400 of these, 1,000 of these, whatever. But in Hardy-Weinberg, I always get it down to like what percentage out of 100, what frequency. So I know that 16 out of 50 have red tail feathers. 16 out of 50 is 32%. So that means these guys, q squared, are 0.32. So q squared is 0.32. If I square root both sides, 0.32 square root, I get like 57. It's actually like 56.56, so I rounded it to 57. So if you kept more digits, your number will be slightly different from mine. So Q is 57.57, and that means this one has to be 43. All right, so if that's the case, then I know the frequency of the red allele. I know the frequency of the blue allele. So red is 0.57. Blue is 0.43. So I know those. Heterozygotes, I just multiply 2 times P times Q, because that's the heterozygote there. So I'd have 2 times P times Q. And again, if you round it a little bit different, your numbers might be very slightly different from mine. 2 times 0.43 equals times 0.57 equals. So I got like 49. For heterozygotes. And then what about homozygous for blue? So that would be P squared. So 0.43 squared. 0.43 times 0.43 equals, so that's like 18.5. That's blue homozygous is like something like that. Seem pretty consistent with what you guys got. Again, you might be off very slightly from me. On the real AP Bio test, they usually accept a range of answers, and then that way if you round one way or another, it doesn't really matter. All right, let's try one more of these, and then we'll be done for today. All right, number 4 there. Try number 4. It says, if 130 out of 200 individuals in a population express the recessive phenotype, what percentage of the population are heterozygotes? Pause it. All right, here we go. It says 130 out of 200 individuals express the recessive phenotype. What percentage of the population is a heterozygote? So I've got the recessive phenotype, so that's good. It'd be a little bit harder if they told me the dominant phenotype was 70 or something like that. So I've got 130 out of 200 have the recessive phenotype. These are alleles. These are genotypes. And if I just use the same colors from the previous question, like blue and red, then the recessive phenotype is this guy, red. So I know that 130 out of 200 are, we'll just say little b, little b. Again, they didn't tell me what letters to use, but I'll just use that since they're already there. And so 130 out of 200, that's 65. So 65% are little b, little b. So Q squared is 0.65. And if I know that, then I square root. So Q is 0.65 square root. We'll call that 81. I got 0.806. And so if Q is 0.81, then letter P has to be 0.19. And then if they want to know the heterozygotes, that's 2PQ. So that's 2 times P times Q, 81. So let's see what that is. 2 times 0.19 equals times 0.81 equals, so I got like 0.31. Again, it was like 0.308 or something like that, so 0.31, heterozygotes. Hardy-Weinberg is more of a steady, sure thing than chi-square. Chi-square, I've said this before, but it's kind of hit or miss. It either shows up big or not much at all, whereas it seems like Hardy-Weinberg almost always has a few points on it. Real quick, just so I don't forget, I missed that part C over here. So back on that question from 2003, the brown-eyed female in the F1 generation resulted from a mutation. Explain what a mutation is and discuss 2 types of mutations that might have made the brown-eyed female. So what would I say if I'm just trying to write a definition of a mutation? What is a mutation? Yeah, it's a change in a gene. It's a change to DNA. So if you said anything about it changing DNA, you got a point. Now, as far as types of mutations, you might have to like think back to your biology 1 class. Anybody give me a name of a type of mutation? These are some of those ones that if it was on multiple choice, It'd be so obvious. It's just pulling them out of the air. What's it? Yeah, a deletion. So let's say I have a sequence like this. That's like A, T, T, G, C, A, C, G, C. Then I could do a deletion. So if I got rid of like the letter A right there, deletion. What else? Do what? Yeah, adding something in. They call that inserting. So insertion, that'd be like adding in a letter. What else? Huh? Yeah, I could switch them. I can invert them. Inversion. So if I took like this little part in the middle, like GCA, and it like cut itself out, flipped around, and then put itself right back in the same spot, you're like, does that ever happen? All the time. So there are many of our genes that are called transposons. They actually cut themselves out and like move to different locations. So they're like genes on your chromosome 12 that used to not be on chromosome 12. They were somewhere else, which is super strange. And you're like, is that very, very rare? No, it's like double digits percentages. How many genes move around? So imagine you have a recipe book, but like some of the sentences for how to make something, they're like moving around. It's kind of bizarre. All right, what else? What is it like if I just trade out that letter A for like a letter G or a letter C? Yeah, a substitution. And again, we wanted 2 of these. There's one more that you might have learned back in your biology 1 class, and it actually like goes along with deletion and insertion. Yeah, a frame shift mutation. Frame shift. This is one that we likely mentioned back in the day, a frame shift. So what is a frame shift mutation? Like, why can a deletion be a frame shift also? Why can an insertion be a frame shift also? Yeah. Yeah, I'm shifting my reading frame. I'm changing the ratios. Remember, we always read in codons, in 3s. So you might recall we had the sentence, the fat cat ate the rat. Well, if I read that, then it's like a triplet. The fat cat ate the rat. If I deleted something, especially early on, like let's say I deleted this H, then now it changes every codon, every triplet afterwards. Now it's tef, atk, ada, tet, her, and then that one's not even a codon. So it changes everything. It changes, it shifts my reading frame. If I had happened to delete 3, and it was the right 3, the fat cat ate the rat, if I just happened to go in and delete like the, that wasn't a frame shift, because I still read fat cat ate the rat. I'm missing that amino acid, but it didn't screw up everybody afterwards. So if I add something in and it's not in a factor of 3, if I delete stuff and it's not in a factor of 3, that type of thing, it can also cause a frame shift mutation. So any of these are fair game for that. If you're in a genetics class, you might get into more translocations and things like that, but that's good for us. So again, these are easy if you just have the words. Like it's obvious what a deletion would be or what an inversion would be. But when you just have a blank sheet of paper and it's like, give me 2 of them, you might be like, I don't know. All right. Any questions on Hardy-Weinberg or chi-square stuff in general or specific? It's hard to know. So the question was, what if I explain it, but I just didn't know the name? Sometimes they're looking for both. And so based off of this, I think, since it says discuss two types that might have produced this, I'm thinking I'm supposed to name them. But I can go and look really quick and just see, hey, back in 2003, what did they want? So I'll pull up the grading rubric and we'll check it out. Let's see, by year, 2003. So I'm going to guess that you did have to have the name of it also. So here's what they said. Yeah. Because they like name them and then I'm supposed to explain them, I'm betting I had to name it. So I would think yes. But if I saw that and I had time and I was like, I can't remember what it's called, I'd be like, there's a type where this happens. There's a type where this happens. So I'd go ahead and do it just in case. But based off of that, I'm thinking that you did have to name it. So yeah, good question. Any other questions? Uh-huh. No. So my correct F2 hypothesis is just that it was we had it as 25, 25, 25, 25, so that's my 1 to 1 to 1 to 1 ratio. So I don't think they said write a null hypothesis. So I didn't have to write, yeah, so I didn't have to write it as a sentence. I could. What I always do is, though, whenever we had like this, it was like 25, 25, 25, 25. That is my expected. That is my null hypothesis. So as long as I have like a 1 to 1 to 1 to 1 ratio, then I can get that point. So let's say, for example, like they say, or 25 per genotype. So some people just say, oh, my hypothesis is I expect a 1 to 1 to 1 to 1 ratio. Cool, we'll take it. Now when you work your problem, you are going to have to use like 25s. Does that make sense? So let's say that I don't know what numbers go in there, but I know it's supposed to be a 1 to 1 to 1 to 1 ratio from like my Punnett square. I got a point for that, if that makes sense. So if I call it my null hypothesis or if I call it my expected, then I'm good. Other questions? Cool. I think that's it for the video. See you guys later.